day everyone, welcome to the course on next generation sequencing technologies, data analysis and applications. In the last class we have started discussion on data formats. We discussed FASTA format and then we looked at the benefits and drawbacks of FASTA format, and we also introduced FASTQ format, right. So, to address the drawbacks of FASTA format, there is this FASTQ format which can combine sequence data along with the quality score. So, in today's class we will be continuing our discussion on this FASTQ format and also look at actual sequencing data. So, this will be the concepts that we will be covering in this class. So, we will discuss FASTQ format in a lot of details, we will talk about ASCII encoding, we will talk about FAST5 format, and these are the keywords you will come across: FASTQ, ASCII and hierarchical data format or HDF. So, as I mentioned earlier, so data from Illumina platform comes in FASTQ format, and we will take an example of this FASTQ format with Illumina data, right. So, this is the FASTA format with quality score or FASTQ. We have the header here followed by the sequence data, there is a spacer plus, then you have the quality score, okay. And these are the reads 1, 2, 3, and if you have millions of reads, this will be there. You have these millions of sequences one after another in a single file. So, this could be quite big files, okay, with all these sequences in place. Now, let us look at the header line. So, last time I just mentioned this is the header line, this contains some information and each header line starts with this @ sign, okay. But what about these other fields that are there? So, you will notice there are multiple fields separated by this colon, okay. So, there are multiple fields there and then there is a spacing here followed by again some information separated by colon, okay. So, we will go one by one, we will discuss what this information actually is, okay. So, the first information, okay. So, this is actually the sequence ID, right. So, what do we mean? What is this sequence ID? This is actually the machine number, the Illumina machine number. So, as I mentioned, the Illumina data comes in the FASTQ format. So, this is actually the machine ID, okay. So, it is D00733, this is the machine ID. Then you have the run number on that instrument, the experiment that is being carried out, right. So, in this case it is the 181st experiment in this machine, okay. Then you have the flow cell ID, right. So, hopefully you remember the concept of flow cell, right. So, all the sequencing in Illumina is done on flow cells. So, this is the flow cell ID and every flow cell has a unique ID. Then you have the lane number in flow cell. If you remember the figure, you have this multiple lanes, right, and this is the lane number in that flow cell 8 here, okay. The next three fields, they are actually showing the positions of this read in the flow cell, okay. So, you have the tile number 2210 here, this is the tile number in the flow cell. Then you have the x coordinate of the cluster in the tile, right, where the cluster is present, and this is the y coordinate of the cluster in the tile, okay. So, you remember the cluster generation process, right. So, x y coordinate gives you the cluster location on the flow cell, right. So, why is it important, right? Why do you actually store this information? So, in general, if the data is good, right, we do not have to worry about this, but if there is some sort of issue, then this information becomes useful, okay. So, we will see this, right, when we look into the data quality check or quality control, okay. So, this gives you the last three numbers, these are the positions in the flow cell. Now, you have the other part, right. So, the first part is about the machine, the location in the flow cell ID, etc. The other part is actually information about the read now, okay. So, this one is actually the read number. Now, this one can mean two things, okay. So, if it is 1, it could be coming from single end sequencing data. So, we talked about this, we are sequencing DNA fragments from one end only, right. So, this is where we have single end sequencing data and the read number will be given as 1, okay. But in case of paired end sequencing, where we are sequencing from both ends, read number 1 would mean this is the read 1, right, only pair of like one of the pair, okay. and then this means for paired end data you can have this number 2 as well. Okay. So, in paired end data, so you can do paired end sequencing in Illumina platform, right. So, you have two files, you get two FASTQ files which will have exactly identical sequence identifiers, but they will have different read number. Okay. So, this field that I just mentioned, rest of the things will be exactly same because they are coming from the same flow cell, same location, same time, etc., same machine, only the read number will be different. Okay. So, let us look at this paired end data, right, how it would be different. Okay. So, here is an example, I am just showing part of the read data. So, that is why the length is different, otherwise the length would be identical. Okay. So, here this is the first of the pair, right. So, sometimes we call them as mates also. Okay. So, this is mate 1, mate 2. Okay. So, this is the first of the pair where you see the header line, okay, and the read number is given as 1. Okay. And then you have the second one, second data, this comes from a separate file. Okay. Remember, you will see, you will get to see two files, okay, which will say, right, some name read1.fastq and the other will say the same name read2.fastq. Okay. So, read1, read2 fastq, this will also be reflected in the read number here. This read number will be reflected in the file name as well as in the header line. Okay. So, here is the header line number is different here for this one, the first read you see this is shown as 1, for the second one this is 2, read 2, right. So, this tells us, right, we are dealing with paired end data. Okay. We have two files, we have these read numbers and we are dealing with paired end data. Okay. The next field, this shows whether the read is filtered or not, right. So, when you are running some experiments in the instrument signal processing unit, right, the software that processes signals and calls the bases, it can do some filtering, right. It has some sort of you can set some filtering, and if the read is filtered means it has some issues probably. N means it is not filtered, right. So, it is actually of good quality as per the software, whatever the thresholds etc. are. Okay. The next part is actually the control number. Okay. So, this is something that is showing whether there are some control experiments that were run in this experiment, right. In some cases you want to do some control for some tests, right, but in this case there is no control experiment here. The last bit here, it actually shows the sample number or usually the index sequence. Okay. So, you remember this index sequence is there when you add the adapters. Okay. So, during adapter ligation you can have this index sequencing, right, and this index sequences allows you to add multiple samples in one experiment, right. So, which we call as sample multiplexing. Okay. So, this gives you the index sequence. Okay. So, just to summarize again in one single table, right. So, if this is the header line, right, you have this information there. So, you have this instrument ID, right, this D00733, then you have this run number on that instrument, flow cell ID, the location here, right, this lane number, tile number, x coordinate, y coordinate, then you have the read number as we have discussed, right, for single end and paired end, what would be this number, right, they will be different, whether the read is filtered or not, whether you have control experiments, right, and finally the sample number which is usually the index sequence. Okay. So, the next line is the actual DNA sequence, this is the sequence that we are interested in, and we have the quality score. Okay. And in between we have the plus sign which is the spacing. So, sometimes you will see this followed by this plus sign, you will again see part of the header. Okay. Now, focusing on the quality score, right. So, this is not number, sometimes you see these numbers like 3, but most of them are like some letter, right, A, C, B, F, G, etc., or some symbols, right, for example, question mark or greater than sign, etc. So, how are these quality scores represented here, right. So, one thing that you should notice is that the quality scores, right. So, this quality score B here is for this base A, this quality score at the rate is for this base T, right. So, you have this one-to-one correspondence, right. So, you can just simply read the quality score down here. Okay. So, this is something that is very convenient for in first key format. Now, the question is how are these quality scores represented, right. So, what do these numbers mean? Can we extract this information and can correlate this with the probability of error, right, that we have discussed in the last class, right. So, probability of error and the accuracy calculation, can we do that from this? So, it turns out these quality scores are ASCII encoded. Okay. So, what is this ASCII encoding, right. So, these numbers, so here are the numbers in this column, they are represented by some sort of symbols or letters. Okay. This is the encoding that we use. Okay. And similarly, we have these 4 columns, right. So, for numbers from 0 to 127 and you have these corresponding characters. Okay. So, these characters can represent these numbers and this can help save space, right, when you are representing these quality scores. Okay. Now, one of the things probably notice, right, only few numbers would be usable for us, right, and this is actually after 33, right. Before 33 we have again the same problem, we are using 2 letters etc., and then we run into the same problem as 2 digits. Only after 33, right, we can use these encodings, okay, and up to 126. Okay. So, this is the range which you can run, right. Now, the problem is the quality score comes from 0 to 40, right. We said it can go higher than 40, but usually in NGS data sets it is probably maximum is 40. Okay. So, how do we represent that? So, what we know is that Illumina uses something called FRET plus 33 encoding, okay, but it earlier used FRET plus 64 encoding. Okay. So, what is this FRET plus 33 encoding? So, the idea is very simple, right. So, if the FRET score, the quality score that comes out of the machine is 25, you just add 33 to that. Okay. So, here 25 plus 33 which is 58 and simply look at the ASCII encoding for 58, okay, and which is colon. The score range that is available as I mentioned is between 33 and 126. Okay. So, if your quality score from the machine is 0, your FRET plus 33 encoding value would be, so FRET plus 33 value would be 0 plus 33 which is be 33. So, encoding would be this question mark exclamation mark, right. Similarly, you can calculate for each quality score what would be the FRET plus 33 value and then you can use the corresponding encoding. Okay. On the other hand, if you see the encoding, right, for example, you saw this, for example, this capital G, okay, in many of these data sets, right. So, for many bases it was G, right, you had this encoding G. So, G now corresponds to 71, right. Now, if you want to calculate that the actual quality score, this will be 71. So, the actual quality score will be 71 minus 33 in FRET plus 33 encoding, right. So, this number you can calculate now, right, and based on this you can calculate the probability of error and the accuracy, right. So, that we discussed earlier. Okay. So, now we have a way to do the encoding or get the quality score from the encoded value. Okay. Earlier Illumina used FRET plus 64 encoding. So, in that case the range was different, right. So, the FRET score, if you take the same example of FRET score of 25 in FRET plus 64 encoding, you will get 25 plus 64, right. So, which will come out to 89, right, and then ASCII encoding for 89 would be capital Y, okay, and the score range that was available in FRET plus 64 was 64 to 126, right. So, if you have quality score 0, your FRET plus 64 value would be 64, right. So, that was the lowest value that was available. Okay. So, now we understand, right, how this encoding works, right, and we can figure out what the Q value actual Q value is, right, if we know the encoding. Now, there is something else that is also added along with this encoding now. Okay. So, what Illumina does now is it does something called quality score binning. Okay. So, what is this quality score binning? So, it simply says like instead of having every possible quality score, why not just give a median value or some sort of representative value for each bin of each range of quality score, right. So, the entire quality score can be divided into, let us say 8 classes here. So, in this example it is 8 classes or 8 bins, right, from 2 to 9, then 10 to 19 etc., and then final one is greater than 40, right. And for each of these bin, why not just give one quality score value. So, every base that falls within this range, it will be given a quality score of representative of that bean, right. So, for example, we can take this example here, right. Let us say any base that has this quality score between 35 and 39, the quality score will be given 37, right. So, all bases will be given the quality score of 37, right. So, there is some sort of change, and this actually helps in also saving space, storage space, it turns out. So, again without going into too much details, you can see that how it will, you can look at Illumina's data, etc., how this helps in storage space, right. So, because of this quality score binning, right, in the earlier data you will see these kind of numbers, question mark, A, C, B, C, etc., all sorts of letters, etc. But in newer data, right, in current data, if you look at this quality score encoding, you will see only limited number of letters, okay, not all possible letters that are out there. Okay. So, for example, in here you see, for example, this hash, F, you see colon and comma. Okay. These 4 letters you see in this quality score because of this quality score binning. Okay. So, most of the bases will appear similar, right. When they fall in one range, they will be given the same value. So, that is for the Illumina data format. What about the Pacific Biosciences SMRT sequencing data format? Do they use the same FASTQ format? So, the answer is no, right. They have different data formats, and we will discuss, right, how this looks like. Okay. So, this SMRT sequencing, it reported the data in HDF5 format earlier. So, we will talk about what is this HDF5 in a moment, and the base call data was present in HDF5 format. Okay. But currently they use something called BAM format, and we will again talk about what is this BAM format when we talk about the mapping problem, etc. Okay. So, BAM format is a machine readable format, it is a binary format, and this BAM format can be converted to FASTQ format. Okay. So, there are tools that will allow us to convert this BAM format to FASTQ format, right. So, why are we interested in getting this to FASTQ format? Because most of the tools are developed for FASTQ format, right. So, we have mentioned that most of the sequencing today is done on the Illumina platform. So, most of the tools that are out there, a lot of tools out there, they are designed for handling FASTQ data sets. Okay. So, this is something that will help, right. If we can convert this BAM format to FASTQ format, you can use those tools. Okay. So, there are 2 tools just mentioned here, one is BAM tools, and the other one is the Pacific Biosciences own tool, which is called the BAM2FASTX. You can download this data from their web page, right. In this web link is given. Okay. Now, what you will probably see if you are working with this SMRT sequencing data and you have sequenced recently, right, in all current PacBio sequences, like if the systems that we have shown, right, in the last class, few classes back, is SQL, SQL2, SQL2E systems, right. The quality scores by default, they will be set to 0, and the SK encoding for all of them, right, will be exclamation mark. So, as you have seen in plate plus 33 format, SK encoding is exclamation mark, right. So, if you download the data and you see the quality score of all bases, this will be given as exclamation mark. Okay. So, this is something what we see, and apparently quality score data cannot be computed reliably for raw data from SMRT, and they are not relevant, and what PacBio has been suggesting, it is actually better to look at the sequencing data itself rather than the quality score. Now, moving on to the ion torrent data format, right. So, how does the ion torrent data looks like? So, the raw data in ion torrent is stored in DAT files. Okay. So, ion torrent raw data means you have the current data, the pH change, right. So, you have this pH change that will induce more current, and that data, that signal is stored in this DAT files, and from that you get the sequence data in either BAM format, FASTQ format, or VCF format. Okay. So, do not be overwhelmed by all these formats, we will discuss these later on. For example, BAM will discuss the human readable version of BAM, and we also discuss the VCF format briefly when we talk about the mutation data analysis. Okay. So, FASTQ data, again, if we can get the data in FASTQ, we can use all the tools that are available for Illumina data analysis, and here in the FASTQ format, the quality score follows plate plus 33 encoding. So, again, you know the actual quality score, you know the actual error rate, you can calculate it yourself. and then you have the nanopore sequencing data format, right. So, here the data comes in something called the FAST5 format. Okay. So, this is a special form of HDF5 format, okay. And how does it look like? Okay. So, before that we can go a little bit into the HDF5 data format. Okay. So, as you probably seen, like in many cases we have this HDF5 format, right. We have seen in case of SMRT, right, there is some earlier it used HDF5, right, and here also we are seeing this FAST5, which is a variant of HDF5. It is a simplified version of HDF5 actually. Okay. So, the name HDF5, it comes from hierarchical data format. So, HDF, and this is the version 5, and you can also have the earlier versions HDF4, etc. And this is used, this data format is used to store very large complex data sets. Okay. So, it is not just for storing this next generation sequencing data, it can be used for storing other different types of data sets. Okay. And the strength of this data set is that you have the metadata included in the data set. Okay. So, you have the data set and you can have as many data sets as you like, and for each of the data set you can also have the metadata saying what kind of data it is, or maybe the condition under which it was collected, etc., etc. Okay. So, it is a nested data format, which means you have groups, okay, and this contains data sets or other group. You can have data sets, this is actual measurements of data, and you have attributes for groups and data sets. Okay. So, what are these terms actually? Okay. So, you can think of this nested data in terms of file system in Windows. Okay. So, if you have these file systems, you have this directory, right. Under this directory you can have a file or you can have a subdirectory, right. Under the subdirectory you can have, let us say, 2 files and another 2 subdirectories, right. So, similarly this is like that system, right. So, you have these groups, right. You have one group, you can have one data set under that group and another group under that group, okay, subgroup. So, this can propagate, and under each group you can have these data sets, multiple data sets, right, as you wish, right, and you can classify them according to the experiment, according to the data collection method, etc. The data sets are the actual data, right. So, for comparison with the Windows file system, these are the files. Okay. So, these are the actual data sets, actual measurements. Okay. And then attributes are like features. Okay. So, for each of these data sets or groups, you can add features saying, okay, this data set was collected for this condition, this data set was collected for this condition, etc. For group of data sets, right, when you define groups, for these groups you can say, okay, for this group of data was collected from this location perhaps, right. So, that kind of thing. Okay. So, as you can see, you can store very complex data sets along with the metadata in here, and this is a data format that is widely used. FAST5 is a specific structure of this HDF5 data. Okay. So, it is a simplified version, it is not as complex. So, what you have is something called raw data and analysis data in FAST5. Okay. So, what is this raw data in FAST5? So, if you go back to the nanopore sequencing principle, right, the nanopore sequencer, it uses current, right, to determine the base sequence, right. We have these nanopores through which the DNA molecule passes, and as the DNA molecule passes, it disturbs the current, right, depending on the base compositions or the KMRs, right, that passes through the nanopore. It gives a specific pattern which can be detected, right. So, the raw data is the values of these currents in picoampere, right. These are very small currents, and this raw data is stored at different time points. Okay. And then you have the analysis data, which is the base call data, right. So, we apply different algorithms or the neural networks, right, artificial neural networks that we talked about on the raw data to detect the base sequence, right, the base KMRs, right. So, this analysis data, it contains the base call data. Okay. So, this is something that is now standard, right. So, you have the raw data in there and the base call data is also available. Now, why is this raw data important? So, for many applications this raw data is useful because it contains the real time data, right, how the current is changing in real time, and that data can be utilized for specific applications. Okay. So, these are the references that we have used. Just to conclude, so we have talked about the FASTQ data format in details, right. So, this is a data format that stores sequence and quality score in one place, right. So, that is a very efficient way of storing data, because this helps in parsing the files very easily, right. We do not have to go back and forth between one or two files, right. So, between this sequence file or the quality file, right. So, which was the case in case of 454, right. You had to really struggle between these two files when you are writing your own program, right. It was time consuming, it took a lot of time, and especially when you are dealing with a huge amount of data and maybe you are dealing with multiple such files, this could become troublesome, right. So, this is something that needed taken care of, right. So, that was taken care of by this FASTQ data format, where you have the sequence and the quality score in one place. Okay. Then we also saw is not just the quality score in one place, right. The quality score is also ASCII encoded, okay, and this again helps in saving storage space, right. And we also saw the quality score binning in Illumina data, which also saves storage space, right. So, earlier Illumina data, it used all sorts of ASCII encoding, right, all possible ASCII encodings for all possible quality values, but now it kind of beans the data into certain number of beans, specific number of beans, it could be 8 or maybe less, and then it assigns a representative value for the quality score in that bean. Okay. So, this also helps in simplifying the data analysis, and it also saves on storage space. The current ASCII encoding that is used by Illumina follows FRET plus 33 format. Earlier data used FRET plus 64 format. So, this is something you need to be careful about when you are analyzing Illumina data. Okay. This will come into play when we are looking into the data analysis part. Okay. So, if it is FRET plus 33, you need to then it is okay, you need to be aware of that, but if it is FRET plus 64, again you need to be aware of that, that you are following FRET plus 64, right. Otherwise, you will kind of overestimate the quality. Okay. So, this is something that will be mentioned when you download a data from the archives, right. Earlier data sets, when you download them, this quality encoding will be mentioned along with the data set, whether it is following FRET plus 64 or FRET plus 33, right. So, specifically it will mention when it is FRET plus 64. And finally, we saw that nanopore data is stored in FAST5 format, and this is a variant of HDF5 format. So, where you have the raw data, which is the current data, and this raw data can be useful in case you have some applications where you want to look at the real time events that are happening, right. So, this is something that is there in the FAST5 format, and then you also have the analysis data, which is the actual sequencing data, right. So, this sequencing data will give you the actual sequence, right, and from the raw data you can also extract the quality score, right, because given the current etc., the algorithm, the base call algorithm can assign the quality score. Now, one of the things that probably we have not discussed, right, how do we process this HDF5 data etc., we will talk about that in data analysis part, right. For FASTQ we can read them quite easily, right, you can open with an editor and you can read one after another, right, one read after another. FAST5 we have not discussed, we can discuss later on how do we process this HDF5 of FAST5. Okay. With that, I will close. Thank you.